Well, I'm a physician trained in hematology, and I've spent my entire professional life doing biomedical research. One of my major interests is the biological activities of bacterial endotoxin. And that sort of background is what led me to go to the Marine Biological Lab in the 1960s to study the horseshoe crab. So I went to the MBL in order to see what the similarities and differences were between amoebocytes and human platelets. And if you want to study blood cells, you have to keep the blood from coagulating. And much to my distress, none of the standard anticoagulants in use at that time prevented limulus blood from clotting, and I was really stumped, and in fact my work came to a halt. And I was trying to figure out what possibly could cause it. By good luck for me, at the same time at Johns Hopkins Hospital, which is where I was a hematology research fellow, I was studying the effects of bacterial endotoxin on blood coagulation in rabbits. Rabbits were a very commonly used model to study bacterial endotoxin because they're so sensitive. And because of that background and my inability to keep limulus blood from clotting, I wondered whether bacterial endotoxin could be the trigger for limulus blood clotting. And when I prepared glassware, which was endotoxin-free, the blood did not clot. And that was the so-called eureka moment when I realized that bacterial endotoxin was a powerful trigger for blood clotting in limulus. Sort of amused that limulus is always described as a primitive animal. It's only primitive by virtue of being the oldest living fossil, but it's not primitive biologically because its ability to control a bacteria that gets into its bloodstream is far superior to ours. The reasons for not using live animals, I think, are apparent to all, and there's increasing pressure not to use live animals if you don't have to. Fortunately, we have recombinant factor C, which has come along, I guess, at exactly the right time, so that we have a choice. We're in a stage where recombinant factor C is working. For people, there are tests that indicate that it detects a wide range of bacteria, which again is not surprising because factor C, it's factor C. And I must admit, I predicted that the test would not be sensitive enough based on recombinant factor C because now you no longer have the benefit of the enzymatic amplification. But according to the specs that are being published by the commercial sources of recombinant factor C, it seems to be as sensitive. It has already been accepted or is about to be accepted in the major pharmacopoeias in Europe and Japan. The FDA has approved it as a substitute, which makes it easier for drug companies to start to use it. A lot of people would think probable that the recombinant factor C is going to totally replace the limulus test. So I think our factor C assay is here to stay.